Good morning and welcome to this wonderful education session. Some in the audience are either directly affected by GIST or have family members by GIST, and some by other sarcomas that might be related to GIST or somewhat different from GIST. And in the course of the morning, although the focus of course is on GIST, we will attempt to draw parallels with advances, how these models can be used to pave the way for other sarcoma types. By the way, I'm Jonathan Fletcher. I run one of the sarcoma research laboratories here at Harvard. My start was in medical and pediatric oncology at the Dana-Farber, but I'm now primarily 100% laboratory based. In fact, I started my fellowship the same year as George Dimitri, my colleague clinical colleague who is co-organizing the conference. So the GIST Cancer Research Fund, about which we'll say a few more words later this morning, has been so remarkably essential to the groundbreaking progress that various laboratories here and at other centers around the country have made in GIST, and frankly models that, as I've hinted, have extended to progress in other sarcoma types. So it's a thrill and an honor to be able to co-organize and participate with you in this conference today. We have an ambitious schedule. This used to be a kind of impromptu talk where George would say a few appreciative words and give a sense of direction, and I was reminded this morning, looking at the schedule, my gosh, everything that George predicted in the previous years, this looks like a good therapeutic lead. This, I think, is going to go somewhere. It's all gone to clinical trial, and there have been insights from those trials. So I think one of the reasons that I'm so delighted and appreciated to be part of this research organization is that it's not idle chatter and thought at the research end, but when we see something that's good, it goes through all the way, from a laboratory concept to a clinical trial, and always, even when they don't work out, with a feedback that enables us to do better the next time. So, we will do our best to continue that process, and I think you'll have a sense for that in the course of the morning today. But it used to be a quick thing, and now last year we had a quick educational session. This year we decided we wanted to be more ambitious, and so more of you have come. We were in a small room before, we're in a big room before, and I think it's a remarkable and wonderful thing that this sort of educational opportunity and very interactive opportunity is a growing event. So I've said enough. What we're going to encourage, because we have a tight timeline, is that even though we'll look for a few minutes at the end of each presentation for questions, let's make it more interactive than that. If you have a question while the speaker is talking, and I hope the rest of the speakers will agree, please just raise your hand, and let's attempt, despite the size of our group here, to have this more discussion-like in terms of the interactiveness. Of course, there'll be opportunities during the break and during the lunch as well. The other cautionary note I want to make, again, because of the tight schedule, when it comes to our coffee break in the middle of the morning and then lunchtime, we'll all have to run for refreshments and right back here because we literally have about 5 or 10 minutes. And lunch we'll eat sitting in the room here during the final 2 or 3 speakers. Okay, good. So that's the orientation. And we'll start with Dr. Chan Rout, well known to many of you, who will discuss some of the whys and the wheres and the hows of surgical advances in GISTs, the questions that still continue to challenge us, and how some of this also is relevant to other sarcoma types. Thanks, Dr. Rout.
Good morning. It's a pleasure to be here today and to see so many familiar faces in the audience. Let me see here. I was quite pleased to see that there are many individuals here who do not have GIST, and so I actually changed my talk a little bit to talk about other advances, surgical advances that might affect non-GIST patients as well. So I changed my topic to Advances in Sarcoma Surgery 2011. I think that many of the speakers here, most of the speakers here, are used to giving talks to other healthcare providers, and when we do so, we have to spend about half of our talk actually talking about what sarcoma is, giving a lot of background to people who are interested in it but really don't know. And I think something that's very unique about this audience is that all of you know GIST and know sarcoma, and so I apologize. I'm just going to actually sort of dive in directly into some data because you don't really need all that background. You already know that already. So I apologize if it seems a little like I'm jumping around, but I do want to hit a number of different points. So what I'm going to talk about are adjuvant therapy for primary GIST and some of the new data that will be forthcoming in that group of individuals. Surgery for metastatic GIST, where things stand with that. Drug-eluting films and non-GIST sarcomas. This is more for people who don't have GIST but have retroperitoneal sarcomas and other tumors that have a high local recurrence rate. And finally, image-guided surgery, about a new operating room that's just opening here, actually just opened yesterday. So the first topic, adjuvant therapy for primary GIST. Now, as all of you know, the success of imatinib and sunitinib in patients with metastatic GIST led to trials looking at the role of imatinib in patients who had resected primary GIST. And this was the seminal trial that was reported several years ago at ASCO and was published 2 years ago. And in this study, a multi-institutional study, patients who had primary KIT-positive GIST, at least 3 centimeters in size, and had undergone a complete resection were randomized to receiving imatinib for 1 year or placebo for 1 year. And all of you are familiar with the data, which show that this trial was stopped early at a pre-planned interim analysis when the data clearly showed that there was an improvement in recurrence-free survival for patients who were treated with imatinib for 1 year, here in the light blue line, compared to placebo in the dark blue line. When the study was reported, there was still no difference seen in overall survival. There was a crossover design. And follow-up has not been long, so we'll be awaiting those data. The reason I bring this up is that there are 3 additional trials that will be looking at the role of adjuvant imatinib in patients with primary resected GIST, looking at 2 years, 3 years, and 5 years. And these latter 2 trials I want to discuss in a little bit more detail. The Scandinavian German trial is going to be reported at the plenary session at ASCO in just 4 weeks. And this study had a slightly different patient population than the one I just showed. It looked at patients who had tumors that were at least 10 centimeters in size, had either ruptured, had a high mitotic count, were either 5 centimeters in size and had a mitotic count of 5, or had focal metastases and had undergone a complete resection. And this study randomized patients to 3 versus 1 year of adjuvant imatinib. And that's going to be, that data will be presented in just a minute. This is the other study. This is a 5-year single-arm phase 2 study that looks at patients, again, who had undergone resection of a primary GIST. Its nickname is PERSIST-5, which stands for Post-Resection Evaluation of Recurrence-Free Survival for Gastrointestinal Stromal Tumors with 5 years of adjuvant imatinib. It was a phase 2 study. The initial target accrual was 133 patients, but after further analysis, we realized that we could actually get the data with a smaller enrollment. And we just completed that enrollment last week. So the eligibility criteria are a little bit more stringent than for the ACOSOG study for primary non-metastatic KIT-positive GIST who have a significant risk of recurrence, and that was defined as any site at least 2 centimeters in size and with a high mitotic count, or a non-gastric GIST at least 5 centimeters in size. And all patients had to undergo a complete margin-negative resection within 12 weeks prior to starting drug. So that study is now closed, and the hope is to have some data available by 2013 at potentially at the ASCO meeting then. Moving on to my next topic, surgery for metastatic GIST. Now, those of you who have been at this conference in the last couple of years have known that, or have seen me talk about the role for surgery in metastatic GIST and try to make some promises about what kind of studies we could do to try to answer whether or not surgery is of any benefit. As you know, the tyrosine kinase inhibitor therapy has really remarkably changed the paradigm for treatment of metastatic disease, and that has led to an interest in determining whether surgery on top of that adds any benefit. And that's because patients do experience durable periods of partial response or stable disease on drug, that true, complete pathologic responses are rare, and that response to drug therapy is not maintained indefinitely. So we have pursued surgery in some individuals, but we still don't know whether this surgery, which is really the adjuvant therapy here, offers any additional benefit beyond staying on imatinib alone. We had hoped to answer this in phase 3 trial designs, the ones that were in development here and that had actually opened in Europe and in China. Unfortunately, a randomization design has been a bit challenging. This was the design of the European study. Basically, individuals who had metastatic GIST and had response to imatinib for 6 months were randomized to either staying on imatinib or getting surgery within a year of starting drug and then going back on imatinib. The target accrual was an ambitious 350 patients with progression-free survival as the main endpoint. And this was targeted to open in 60 centers in Europe and Australia. The Chinese trial had a slightly different eligibility requirement, but it was also targeted for about 280 patients. And our U.S. study was targeted for about 230 patients. And unfortunately, the European study was closed after only an accrual of 15 patients over about a year and a half. And the Chinese study is also closed. And the problem has been trying to patients often come in with a preconceived idea of whether they either want surgery or they don't want surgery, and very few individuals are really willing to be randomized, and that's understandable when the treatment in one arm is surgery plus drug, and the other arm is drug alone. So we've struggled with a way to still answer this question because we think it is an important question to answer. And the reason is that the complication rates are high enough that I think we need to justify doing an operation by showing some improvement in survival before we recommend it for people if we have high complication rates. So a new approach to try to answer this question is comparative effectiveness research. This is actually a buzzword that President Obama has been using to try to justify some of the treatments that we offer for a number of different illnesses, including cancer therapy. And the way this study, and this is now becoming an interest in a number of the cancer cooperative groups as well. And what this study does is essentially allows the patient who meets the strict eligibility requirements of what would otherwise be a phase 3 study to pick their preferred treatment. And again, the two treatments would be the ones that we were always considering, staying on imatinib or considering surgery and then resuming imatinib. And then as long as they met our strict eligibility requirements, which I'll show on the next slide, that we would essentially follow the outcomes for these individuals. The data analysis afterwards also would include a very robust way of trying to take into account certain biases that may come up with such a trial design. And one of the concerns has always been that depending on what treatment center a patient is evaluated at, may have a certain bias. There might be someone at one center that says, there is no benefit to surgery, you just need to stay on drug. Whereas at another center, we think surgery is very good, everyone should get an operation if they're eligible. And that kind of bias can certainly skew the results. And so there is a propensity score analysis, which is tries to take into account a number of these other factors that might not necessarily show up in a phase 3 design. These are the eligibility requirements for the study. Adults with a good performance status, KIT positive, metastatic disease confined to the abdomen that's considered resectable, and that's again a potential bias. They may have had prior surgery for their primary tumor, but no prior surgery for metastatic tumor. They may have also gone on imatinib either prior to the original resection of the original tumor or after resection of the primary tumor, as long as they're not on imatinib when the metastatic disease was diagnosed. And radiofrequency ablation would be permitted for liver disease and liver disease only, but not for other sites of disease. And so essentially this would be the schema. Again, patients would have to have stable disease for at least 6 months. But then this is where it differs from a typical phase 3 design. Instead of randomization, it's patient selection as to which arm that they prefer to follow. And then we essentially follow until progression, and then we do the propensity score analysis, taking into account the various treatment centers as well as other biases. And in fact, we would analyze the data based on site of treatment as well as outcome. A third topic that I want to discuss is actually more applicable to non-GIST. Actually, let me say one thing about the prior study. We're in the process of developing it. It's a design that we have developed in conjunction with the group in Europe, and we'll be proposing it to the cancer cooperative group soon. As you may know, that they're undergoing a reorganization and consolidation of a number of the groups. For instance, CALGB and ACOSOG are joining forces to become a single cooperative group, and so we're waiting for the dust to settle from that before moving forward with the proposal. Now, this is more of a basic science study that we are doing here in conjunction with one of the thoracic surgeons. The drug-eluting films are an interest for any kind of tumor, especially sarcomas of the abdomen and retroperitoneum, which have a tendency to recur